Everybody, I'm glad to see everyone back here, and I'm pleased to introduce Dr. Tom Hahn. Tom is a professor at University of California, Davis. I met Tom because he was leading sound identification field trips at the Mona Lake Bird Chautauqua, an event that I know some of you have attended. Tom has been involved in research with his graduate students on a wide variety of finch species, everything from evening grosbeaks and rosy finches to the house purple and Cassin's finches, studying things from vocal variation, habitat use, reproductive schedules, and so on. So tonight he'll be talking to us about the natural history of the Western Cardueline finches. Thank you.
Thanks for that, John. I'm going to share my screen and make sure I get everything right here, I hope. Share. And now here we go. So thank you very much for inviting me to participate in this. This is really fun. And what I'm going to talk about here is one of my favorite topics. Actually, I want to preface this by saying I'm actually out on the Olympic Peninsula at a cabin right now. And so if there's bad lags with my internet, Jennifer will just turn my video off and hopefully that will help a little bit. But I actually heard type 3 crossbills and type 1 evening grosbeaks just outside today. So that put me in a good mood to present this. Okay, so I'm gonna talk about finches today. It's birds that I've been studying for a long time. And I'll just start out by talking a little bit about what is a finch, probably something I don't need to tell this audience, but there are a bunch of different kinds of birds that we do call finches. All of them are songbirds. There's phloeciids, there are estrildids like zebra finches, there are emberizids like this black-headed grosbeak, and there are fringillids like this chaffinch. I'm going to focus on fringillids, and actually specifically, I'll be talking about the Carduelines, which include birds like the ones shown here on the right. Even the Hawaiian honeycreepers are derived from a colonizing Cardueline. And the Western Carduelines, in the Western United States, are some of my favorite birds. Here's a few of the ones, most of which we've gotten to study to some degree or another. I actually got started with red crossbills as a graduate student in John Wingfield's lab at the University of Washington back in the late 1980s. And I was interested in them specifically because of their reputed extreme reproductive flexibility. And I was interested in studying that from a variety of standpoints. So that's how I got started. But then I've had the opportunity to study a number of other different kinds of finches and other aspects of crossbill biology. So the things I've chosen to focus on today, excuse me, include feeding adaptations, annual schedules, heterospecific vocal imitation in song, call learning and matching, and cryptic taxa. And I had trouble figuring out what order to put these things in because it always seemed like whatever I was talking about, there was something I wished I had said beforehand, but this works okay. And when necessary, I'll just say, and we'll talk about that later. So we'll start a little bit with bills. Bills are sort of the thing that typify finches. And in fact, I think a lot of people, if you ask them to think of a bird, they probably would think, just imagine a finch and a finch beak. Maybe I'm wrong about that, but finches just seem like sort of the archetypal bird in a lot of ways. Their bills, as kind of mundane as they look, are really good for handling seeds in particular, although this isn't all finches do with their beaks, of course. And they have these husking grooves that are up in the upper bill. And when they can get the right kind of seed in there, it fits just right. And by hitting it with the lower bill and holding it in place with the tongue, they can husk the seeds. And the structure of these palate grooves actually determines which types of seeds, which shapes and sizes of seeds particular finches are good at handling. This picture I actually dredged out. It's one I took when I was like 17 years old at a kids camp that I used to work at in Wyoming. The evening grosbeaks would show up late in the summer. And it was fun to watch them because they would come through and pretty much discard the fruit and eat the seeds out of this sarvice berry that was all over the place there. And of course, evening grosbeaks are good at cracking pretty good sized seeds. Of course, crossbills bills are maybe among the most specialized of the finch bills, if you exclude, the Hawaiian honeycreepers. They are good at hulling seeds, just as other finches are, but they of course have this very cool crossed bill structure, which they use to pry up the scales of cones to get at the seeds. And they put the straight upper bill against the basal scale, and the curved to the left or to the right lower bill goes against the more distal scale. And they bite in between the scales, and then they can, by laterally moving their bill, they have very strong muscles that are asymmetrical on their head to pull the bill, the upper and lower bill laterally apart. And they also turn their head. And I have a little video here that Craig Benkman shared with me that shows this. Many of you have probably seen this video. You can see this individual. It's really quite extraordinary how much he is able to pull the upper and lower bill apart laterally. But you can also see he's turning his head as he does it. It gives him even a little more leverage. Then you can see his tongue coming out to try to dislodge the seeds. In these green lodgepole pine cones, it doesn't work very well because they're sort of stuck down there. So he's kind of hooking it out there, and now he's going to hull it. And there went the seed hulls. And this behavior and this morphology, the combination of these two things, allows crossbills to get at seeds that are protected from other birds. So here's some western hemlock cones. These are the tiniest cones that crossbills in North America typically use. They're only about as big as the last little section of your little finger. And they have very papery scales, and type 3 red crossbills, we'll come back to the types later. Most of you are probably familiar with them, but I'll talk a bit about those later. They can bite between these scales and get these green cones open. And then what they do is first of all, they pull the cone off the tree stem, and then they hold it in their feet and they turn it around and around, and they just pry up the scales and extract the seeds. These 3 cones over on the right are western hemlock cones that are the same age exactly as these ones on the left, but these had been removed and opened and then dropped by crossbills to the ground. If you're out in July in a place where there's new cones developing on spruces, or hemlocks or Douglas fir, you'll often find trees that they are hitting really hard, and these trees will be littered underneath with these opened crossbill cones. And they always look different from ones opened by squirrels, which will have had the scales actually peeled off completely, leaving just the core. Big crossbills like type 2s can actually open green cones, immature cones of things like ponderosa pine. Here's some ponderosa cones in August in the eastern Cascades of Washington. And here's some from the same tree that some type 2 crossbills had opened. They don't actually remove these cones from the tree to get access to them. They just work on them in place. It's a lot of work, but it has a high payoff because these seeds are quite big, and there's many of them in a cone. The differences in morphology from small to very large crossbills are correlated with the types of trees that they are well suited to forage from, especially from the closed cones. And they're, as Craig Benkman has described, the adaptive radiation in crossbills quite nicely. So some other things about, and I'll come back to that actually, some other things about crossbills foraging, and not just crossbills, but Cardueline finches in general, is that many of them have very capacious crops to carry a large bunch of seeds or whatever else they may be eating, and also gular or sublingual sacs. This is a brambling, which is a fringiline finch, and you can see he is carrying all of his food that he's bringing to his mate and to the babies just out loose in his beak, just like one of our emberizine sparrows would do. Cardueline finches, like this evening grosbeak, they swallow the food down into their crop and then or into a sublingual pouch or whatever, and then they regurgitate it. So this female is feeding this fledgling. Here's a crossbill regurgitating seed pulp to his mate, who's incubating or brooding babies. This is from Ian Newton's book. And the crops, if you happen to be lucky to capture any of these finches like siskins or crossbills, you can see the crops. The bird's head is over here and the tail is over here. And my former student, Jamie Cornelius, is just holding the bird gently and pulling the feathers aside. And you can see the crop contents here. Here's the jugular vein going across right there. And the papery thin skin allows us to see into the crop. Here's a closer view. You can see these big ponderosa pine seeds in the crop, which that bird will regurgitate if he or she is going to feed them to another individual. This allows crossbills to feed on things that are not, on food sources that are not just right next to their nest. They can go off quite a distance, collect a large quantity of food, and then take it back and visit the nest relatively infrequently. This is beneficial because it presumably reduces the amount of attention that the nest is drawing. They can visit the nest as infrequently as once an hour to feed, and they just feed a large quantity at that time. And the males feed the females on the nest. This is very common in Cardueline finches. Some of the birds don't have a big crop, but they do have these sublingual or gular sublingual pouches or gular sacs. Rosy finches are a good example of this. Many of you have probably seen rosy finches up in the high mountains of the West. Here's a gray crowned rosy finch near Tioga Pass. And rosies nest in very high elevation places, quite inaccessible, and actually not right next to good food resources. They're up in rock piles and stuff, and they have to fly some distance, often hundreds of meters or even further, to get to their foraging places. And these gular sacs actually facilitate their being able to take a reasonable quantity of food back with their less frequent visits to the nest. Something I want to tell you about, I'm not sure how many of you are aware of this, but it's a very cool ecological interaction between fish and birds like rosy finches. A student at UC Davis named Pete Epanchin, who finished his PhD a number of years ago, worked on this. And in the lake basins in the High Sierra that, like this one, that do not have fish in them, which historically none of them did, or virtually none of them did, they were all introduced fish, trout. There are much greater insect emergences, for example, mayflies, from these fishless lakes, and the congregations of rosy finches in these areas are much larger. They exploit these resource pulses during breeding, and being able to carry large quantities in a gular sac back to their nest that's somewhat distant. It might be way up in the rocks up here, so they have to make flights down to the lake, can be advantageous. This is just a quick little thing from one of Pete's papers that shows that in fishless lakes, there's lots of mayfly nymphs. These emerge as big pulses of mayfly adults, and there's a big increase in the numbers of rosy finches around these kinds of lakes. Whereas in places where non-native trout have been introduced, those lake basins, they still have rosy finches, but not nearly as many. There are not these huge pulses of adult mayflies coming out of those. And actually, things like Sequoia Kings Canyon National Park are working to remove fish from a lot of these lakes. It's not yet known exactly how important these resource pulses are to other types of birds that have different ecology from rosy finches, but another student, Mary Clapp at UC Davis right now, is studying that. And the effects on rosy finches do seem to be the greatest. Here's just a couple pictures from Newton's book. Here's a pine grosbeak with a big load of stuff in its gular sac, and bullfinches also do the same thing. So of course, those are not, bullfinches are not Western North American, but we do have pine grosbeaks. So the feeding behavior and the feeding morphological adaptations that these birds have contribute to some of the interesting features of their annual schedule. So I'll talk about those for a few minutes now. And I got interested in this specifically because of crossbills reputedly being able to nest in any month. At the time I started my dissertation work, the dogma was that they're basically equally likely to nest in any month, and that what they do is wander around until they find a big cone crop, and then they begin foraging on it and start breeding and breed as long as that is available. So I was interested in looking into this in greater detail. And one of the main pieces of evidence people used for this was just that these birds can nest on both the longest and shortest days of the year. And so the argument was they must not be using photoperiod. That turns out to be logically incorrect. You need to know more about photoperiodism. That's a separate topic for a lecture. White-winged crossbills are similar to red crossbills in having these really extended flexible breeding seasons. And even pine siskins and redpolls can start breeding quite early if there's a big cone crop with a large seed supply, and can also, siskins in particular, can extend their breeding late at the very end of the summer into September if there's a large seed supply. All of these birds, I think the main thing that got people's attention was winter breeding. It just really, when conditions are really cold. Here's a couple of photographs of one of my field sites, which actually, I'm sorry to say, may be in flames at the moment. It's in the Rattlesnake Creek drainage east of Mount Rainier. And there's a big fire up there right now. I'm not sure exactly what's burned, but this is an area called Devil's Table. And this is the ponderosa forest in August, when there were lots of type 2 red crossbills foraging there on a huge ponderosa pine cone crop. This is back in 1988. And then in the winter of 1989, this is the same area there, the crossbills were breeding again using the remaining seeds in the cones that had opened the previous September. And it really just is this tendency of crossbills in particular to be able to breed on the longest and shortest days of the year that got people's attention. And people assumed they must be using food as the primary or even only cue to regulate timing of reproduction. And they drew sort of what they thought were logical inferences that breeding must be continuous from when they start in the summer until they stop the next spring on a big cone crop like ponderosa pine. I don't want to spend a lot of time talking about this, but there are papers that have been miscited that didn't actually show what they said. And the bottom line is actually that they're not as flexible as that. I'll come back to that, but they are extraordinary in being able to nest in the winter. And this terrible picture is a scan of a bad photocopy of a paper by Bailey, Needrak, and Bailey, 1953, the Red Crossbills of Colorado, that I got as a graduate student through interlibrary loan. And here, I love this: a heavy wet snow blanketed the nest site, marked with the X. X marks the nest site of a type 2 crossbill. They didn't know the types then. They called them Bent's crossbills at the time. And here she is in another photograph from that paper, sitting on her nest with snow all around it. And she hatched these eggs. And redpolls are actually, and siskins can do this kind of thing too. Here's a photograph that John Wingfield provided me with from up on the north slope of the Brooks Range in Alaska, a common redpoll female sitting on her nest. And in a little birch tree. This is in May, but the weather can become very bad there. This is a very nicely insulated nest with ptarmigan feathers. And then it's very hard to see this here. Hopefully you can. Here's her beak. Here's her little eye stripes, and her eyes are right there. She's sitting in her nest there, and it's all surrounded by snow. And of course, lots of birds are able to do this. The white-crowned sparrows that we've studied quite a bit up in the High Sierra can defend their nests from adverse weather like this, but not to the extent that Cardueline finches do. They are, the females just stay on the nest, the males feed them. White crowns, that doesn't happen. And so the females very seldom need to leave the nest. And so they can deal with these extremely cold conditions and raise their young under winter conditions, in fact. Of course, the weather got better shortly after this for this redpoll, but the crossbills breeding in January in the Eastern Cascades were dealing with cold and snow for weeks during breeding, and they just were using the ponderosa pine seeds, and the males were feeding the females at the nests. So one question, then I alluded to this already, is how temporally flexible are crossbills? They can breed in July, they can breed in January. What is happening with these schedules? It turns out, so we've been looking at this literally for like 30 years now, and we have completely failed to be able to find evidence that they breed straight through the fall the way a number of people have reported, even if they're in places with large cone crops. And here's some data from 4 different crossbill types, 2, 3, 4, and 5 from Western North America. And one of the things I did as a graduate student and a postdoc and early in my career as a professor was I would do laparotomies on these birds in the field. I would anesthetize them and visualize the gonad to see what the gonad condition was. And in the summer, these birds are typically all in breeding condition. But by the end of September, the gonads have collapsed in all of these. And actually, we have lots of other kinds of evidence. The cloacal protuberances shrink and the females refeather their brood patches and so forth. We just find no evidence of fall breeding the way people have reported casually in various kinds of papers. And in fact, furthermore, if you look at this is just type 2, large red crossbills in 2 different years, many years apart at 2 locations. One was at Devil's Table, and one was in Washington and east of Mount Rainier, and one was in the Warner Mountains in Oregon. And this is all the data together from those 2 years. The birds are in breeding condition in the summer. The gonads collapse in the fall. And then they can, in both of these years, it was a large ponderosa pine cone crop, and the birds came back into breeding condition and resumed breeding at the same locations using the same cone crop on shorter, colder days than when they collapsed the gonads the previous autumn. So we just call this the autumn hiatus in reproduction of crossbills. And I'll come back to this in a little while. It's a good question why an opportunist like a crossbill would cease breeding at the very time of year when the cone crop is at its peak for foraging efficiency. September is often when the feeding rates are the highest they're going to be all year. But they do this consistently. And I'll cut to the chase here and say they're molting here, but I'll show you that in just a few minutes. Okay, so more aspects of the annual schedules of crossbills in particular. They're known to track cone crops. This is from Newton's book. This is just showing that spruce cone crops and crossbill numbers track one another very closely. This is in Northern Europe. And there's just a nice relationship between numbers of crossbill pairs and the amount of spruce seed that's being produced. Furthermore, so that's been used as evidence that they just wander around until they find cones, and that's great. They also are eruptive. So one year in Switzerland at a banding station, lots and lots of invading crossbills, common crossbills, also Loxia curvirostra, at least currently, were banded. And then ones that were recovered within that same year were all someplace around the general vicinity of southern central Europe. But in subsequent years, they showed up in way to the northeast in northeastern Europe and Russia. And together, all of this is kind of used as evidence of just the erraticness of the movements of crossbills. But it glosses over something really interesting, which is they are actually seasonal migrants as well. So they move typically in the spring. Here's some data from Jamie Cornelius, one of my former students who's at Oregon State now. And she found that in field caught crossbills, the fat score, subcutaneous fat that you can see on the furcular area and abdominal area, peaks in May and June, which is exactly when these birds typically are moving. And Newton talks about them moving in May and June as well, but we've seen it repeatedly that you'll find moving crossbills moving through habitat often that has no cones and where they don't settle. But they're often, if you catch them, they're often some of the fattest crossbills that you will observe all year. And in fact, the fact that they have greater fat scores then than they even do in the winter is the type of thing that people often use as evidence of migratory fattening, that it exceeds winter fattening. I'm not going to go through this whole thing, but one of the other things that Jamie did with captive birds was find that their activity levels were also highest in June. And this is just perch hopping activity, so it's not nocturnal, but they show more activity at this time of year as well. And their food intake goes up. And I actually found this in an experiment that I did as a graduate student as well. I didn't look at activity, but I found that fattening and body mass and food consumption were up then. So these things are consistent with the crossbills actually having a seasonal migration in the spring. One question that's really interesting to think about is how do they fuel this when they're doing it at a time when conifer seed supplies, even at places that have lots of cones from the last year, are tanking. They're reaching a seasonal or an annual minimum. It turns out that if you go out and observe crossbills in the spring, in May and June, frequently it's very difficult even to find them, in my experience, foraging on conifer seeds. They're mostly eating insects. And if you look at what's in their crops, again, visualizing the crop in June, we don't have any data. This is something we're just starting to compile. But in June, there is a peak in non-seed contents of the crops. Now, we don't know exactly what this stuff is. Some of it could be grit and whatnot, but it looks like insect gunk. It looks, and if you observe them foraging in these times, you often find them foraging on budworms and aphids and things like that. And so, for example, in May, one year, I found big flocks of type 3 crossbills opening up these rolled elm leaves on the University of Washington campus and licking the woolly aphids out of those. Found swarms of crossbills near Mount Hood eating budworms on abies, on firs. They eat a lot of coolie gall aphids, which are these little waxy thingies that are one life stage of these infests the growth tips of Douglas fir, and crossbills eat lots of these things. And then just most recently, I found type 2 crossbills opening these petiole galls on cottonwoods and eating the aphids out of those. And in all of these cases, I couldn't find, there weren't even any cones around for the birds to eat, but the birds were abundant and moving through the area. So I think one of the things from a natural history standpoint that is the most interesting to me about this is just that these birds, they are clearly adapted to being specialized on conifer seeds, but I don't think they actually could be, the nomadic ones, if they did not have a food source like this that could tide them over during their movements from one cone crop to another, since cones don't consistently get produced at the same locations in each year, in consecutive years, in most of the conifers that these birds use. They need something that's going to allow them to fatten and or just fuel and root while they move through unsuitable habitat looking for a new place to settle. So I think these insect foods are super important, and it's been neglected. It's something that would be really good to get additional information about. So the last thing I'll talk about on annual schedules is molt. And the question basically is, what is happening when the gonads collapse in late summer and autumn? Why would the birds do that? Well, it's because they're molting. This guy here has 4 new or growing primaries, and then 4 old ones. And number 5, you can't even see here. It's a pin or a small sheath feather right in there. So he's in the middle of molt. This was a bird in Wyoming. And this just shows it better. Crossbills do a nice, fairly standard molt. It's spread out, and they do have some weird stuff about it, but pretty much once they get started in the middle of the summer, they usually proceed pretty normally and accelerate as when they get about halfway through the primary flight feathers, they accelerate into the body molt. Here's a male that was formerly kind of yellowish orange, who's putting in a bunch of nice bright new red feathers. So his next plumage is going to be very red. And Jamie Cornelius plotted up the molt data that we have, and you can see typically it's concentrated in the late summer, and it accelerates, the speed of it accelerates in September so that they're finishing in October, and by November they're finished. This is exactly when the gonads collapse, and after that's finished, they are able to come back into breeding condition a month or so later. So here's what I think the crossbill year actually really looks like. On the outer circle here, we have sort of the conifer development, and the cones are developing, new cones are developing someplace in June and July. By August, they're maturing. By the end of August and into September, they're maturing, and the cones are opening and becoming really easy to get the seeds out of. But crossbills can start harvesting seeds certainly by the beginning of or middle of July from most of the trees that they use. And then seed fall varies a lot from year to year and among tree species. But that is going to determine whether there's an adequate seed supply to sustain them going into the subsequent months. The seasonal nomadic migration is what's going to bring them to a place where there's new cones developing, and they can settle, and then they can breed in the summer. They also begin the molt partway through breeding and finish it by late October, beginning of November. If the seed supply remains good, then they can resume breeding facultatively in winter and into the spring. And then they would go into another seasonal nomadic migration. If there's a collapse of the seed supply, or they never find a good cone crop, they will be forced to engage in a facultative eruptive or a facultative nomadic movement, looking for some place that they can survive at the least, if not breed. So basically, this is when the autumn hiatus is in the late stages of molt. And that's what we've been observing and in terms of the timing of gonadal collapse. This schedule is fairly typical for a bunch of different Carduelines, I think, especially the more flexible ones like pine siskins can be like this as well. To one degree or another, they may not have quite the degree of flexibility in timing. Siskins, for example, probably don't begin breeding before March in the Rocky Mountains or something, but crossbills can start in January. Okay, I'm going to change tacks here pretty dramatically here and start talking more about vocalizations for a little while. It's something that I haven't really studied super intensively myself, but I've paid a lot of attention to it because I just like bird vocalizations. And when I started learning about birds back when I was a much younger fellow, the dogma was that in songbirds, song is learned and calls are innate. And actually, way back in the 1960s, Paul Mundinger showed in Cardueline finches that calls are plastic in adult birds. And then, so we'll talk about that a little bit. He started out with American goldfinches, and he showed that there was matched pairs. So there's males on the top, females on the bottom. These are the call forms that these pairs of males and females had. He showed these matched calls. Fred Sampson showed the same thing in Cassin's finches. Here's I don't know which is the male and which is the female, but one or the other. So let's say male, female, male, female, male, female, male, female of 4 pairs of Cassin's finches in Utah. And actually, they both did experiments too, showing that the female is able to recognize her mate's calls, and she will call from the nest or even leave the nest to be fed if you do a playback of the mate's call, the mate's pair-specific call, but not just the call of another male of the species. Mundinger also showed that this wasn't just pairs choosing mates that match their calls, just sort of assortatively mating with ones that happen to sound like them. So this is actually a European siskin male. And what Mundinger showed experimentally was if he took this male, and here the male has these different, I don't know what the vocal repertoire of these European siskins is, but they have multiple call note types in their repertoires. Here were 3 different note types that this male had before he was placed into a flock of unfamiliar siskins. And then afterwards, he had replaced each of these with these new versions, which matched models among the individuals in that group. So he had modified his calls as an adult to basically conform to the new social group that he was in. My former student, Kendra Sewell, did her dissertation on this with red crossbills, and she showed pair matching. And it's going to take me just a second to explain this figure here, so bear with me. So what she's got here on the top is a pair of birds. This is a type 4 red crossbill and a type 3 that she put together in an aviary. They formed a pair. This is what their calls looked like before they were paired, and then some number of weeks after they had been living together for quite a while. And then down here, she did it with a pair of type 3s. These are both type 3s. This is before and after. And what she's plotted here is essentially where their calls are in acoustic space, in multivariate acoustic space. And the open circles are one of these birds before before pairing, and the closed circles are where that bird was after pairing. The closed squares are the other individual, and the open squares, and the closed squares are that bird after it's been with its mate for a while. And you can see they both shifted. This one shifted there, this one shifted up there, but they didn't converge really. So these birds are of different call types, and the calls are still of different call types after they've paired. In this pair of type 3s, they actually started quite distinct, and they both converged in a particular area of acoustic space. So this is what she looked at this in a bunch of different individual pairs that she set up, with ones that were same type individuals versus different type individuals. And what she found was that the same type individuals tend to converge, especially if they like each other and form a pair. And different type individuals may try to do so. This call definitely looks different than it did before, and so does this one. But they have a hard time converging on the other call type. Craig Bankman and his students have found occasional cases where they will shift, where birds will shift to a new call type in the field, but Kendra didn't see it happen in her captive birds. Kendra also found that the babies, Jeff Groth described this with in his monograph from his dissertation, but it was anecdotal and just based on a few birds, and Kendra followed that up to show that young birds, young crossbills that are cross-fostered to other individuals. She was breeding pairs in captivity, and then she had foster parent pairs. This was a monumental task to do for her dissertation. She showed that the young birds acquired the call types of their adoptive parents. So these actually, we didn't know about type 10 at the time, but these actually look like type 10s. These were type 4s. So the biological parents were type 4s. The adoptive parents, we thought, were type 4s. We've subsequently learned that these rising calls that don't have this dropping component are actually a different call type, but this bird converged on the adoptive parents. These were its biological parents. And here's another one that acquired the call of one of the adoptive parents, which looks like a type 10, and the other adoptive parent was a type 4. But the biological parents were type 3s. So she did this with a bunch of different individuals, and she was cross-fostering to same and foreign call type, and showed that they acquire the calls of the adoptive parent. And again, Jeff Groth showed this, and it's in his dissertation, but it was based on just a handful of individuals and wasn't really controlled properly. And he acknowledged that, just said it needs to be followed up. So the babies learn the calls very readily of a foreign type from what their biological parents' calls were. But once they've crystallized a call type, adult crossbills, they're able to converge on other individuals of their own type, but it's much harder for them to shift call types completely. Okay, I'm going to say a little bit about heterospecific vocal imitation in song, and I'm just going to go through this pretty quickly. As usual, I'm taking longer than I intend. This was something I blundered into, and I'll just put a shout out for doing observational biology in the field. I love it, and I know most of you probably do as well. And this is where we get our raw material for asking questions and coming up with hypotheses. This bird here, this is a recording of a Cassin's finch that I made near Devil's Table, one of the sites that I showed a picture of earlier. And these are all type 4 red crossbill calls in that song. The songs of these finches, yeah, I'm not going to go into more detail about the song structure. There's a couple different forms of song, and this song form has lots of heterospecific imitations in it. But I kept getting fooled. I kept looking for the crossbills when I would hear these type 4 crossbills, and frequently it would be a male Cassin's finch singing. And I finally paid more attention and discovered there were even grosbeaks in there. There are flickers in there, and various other things. Here's a song. Hopefully you'll be able to hear this. Oh my goodness, I can't remember if I clicked on those things. I think I did. But hopefully you'll be able to hear this. Here we go. Here's a Cassin's finch song. Lots of heterospecific imitations in this part, and I pulled just a few of them out. That's pine grosbeak. That's Townsend solitaire. So I'm hoping some of you can hear this well enough that you can test yourselves. That's a robin peak call. And that's a type 1 evening grosbeak. Cassin's finches are not the only ones that do this. These imitations, by the way, are really good. Here's a lesser goldfinch. And this one, just pulling a few out. Here's type 2 evening grosbeak. So that right there, and this. And then there's a trill of an evening grosbeak. To my knowledge, we don't know yet whether the trills are type specific in evening grosbeaks, and I'm going to come back to the types in evening grosbeaks in just a second. There's a killdeer. Here's a hairy woodpecker. And here is part of a western wood pewee. We still don't know what this is for. It's presumably some kind of sexually selected thing where females like this, but it could be that the females like a larger note repertoire that's selected for this. It could be that they actually pay attention to how well the males imitate the model sounds. That's certainly what female sparrows do. They prefer males that do good copies of the songs that they were tutored with. So it's unclear. It's also not known yet how these birds acquire these. Do they acquire them from each other after one of them learned one of these things? So a lot of them are passed from one finch to the other. Or are they all copying them from the original, from the model species? We don't know that yet. Okay, the last thing I'm going to talk about just for a couple minutes is cryptic taxa in these birds. And we already mentioned the adaptive radiation of red crossbills that Craig Benkman has studied extensively. Jeff Groth was the one who described this in his dissertation that he did from the Museum of Vertebrate Zoology. at Berkeley. And he actually studied them as a master's student at Virginia Tech with a guy named Curtis Adkisson, who's the first person I ever heard actually talk about call types and crossbills. Jeff described 8 different vocal types. Here's 5 of them with various aspects of the sizes of males. He described 8. There subsequently were 2 new ones described, 9 and 10. 9 has become the cassia crossbill, a separate species. 10 is the one that Kenneth Irwin described from the north coast of California. And those turn out to be up and down the coast of the west coast as well in Sitka spruce. But there also, the crossbills aren't the only ones. The evening grosbeaks have these call variants as well. And I'll just play a few for you. Actually, just briefly, this is something that I noticed when I came back to California after living in the Northwest for a while, and I had been hearing these all the time. That's what we call a type 1 evening grosbeak. It starts high, rises, drops, dwells for a moment, and then drops again. It's a very clear, loud cheer. And then this is what I was hearing in the Sierra Nevada. And they just didn't sound anything alike to me. It's easier if we slow them down to hear the differences. That's type 1. Here's type 2. And then one of my students, Rod Kelsey, and I were in Colorado, and we blundered into a bunch of grosbeaks. We were looking for crossbills, and they sounded like this, and we immediately realized they were something different still. And here that slowed down. You can hear the big bounce in that. These, I'm not going to play all of them for you. So basically, my students, Kendra Sewell and Rod Kelsey, dredged together a bunch of stuff and produced a paper on this. And type 1s have this typical structure. 2s, 3s are from the east. 4s were from mostly the southern Rocky Mountains. We didn't have a whole lot on that. And then a guy named Rich Hoyer, who I'd never met, but he's definitely up on evening grosbeaks. He had a recording of what presumably was yet another type. It's distinct from, and he kindly let us use that. It's just a single individual. And these types turned out to be distributed around in a way that matched pretty nicely Grinnell's 100-year-old description of 5 different evening grosbeak subspecies. And so what the AOU currently categorizes as Brookside, the western evening grosbeak, has what we think are 2 cryptic taxa hidden in it that Grinnell called Californicus from the Sierra Nevada and extreme southern Cascades, and Warreni from the southern Rocky Mountains. So this deserves a bunch of additional attention as well. Here's a new map that Matt Young shared with me recently. And I'm going to say something about Matt in just a second. So there's more to this. Matt Young recently told me about purple finches. He was convinced that purple finches had eastern and western call forms. And I've looked into it, and I agree with them. The eastern ones they're very short and hard to resolve. But once you listen to them, you can hear that the eastern ones are very monotonic and the western ones rise. And this is something that we're looking into. We need more good recordings of just the little soft flight calls of purple finches. Okay, so I'm pretty much done, and I'm almost going to finish on schedule, it looks like. And I just put this in here because I just love the looks of baby crossbills. This is from Ian Newton's book. And these little guys, they just sort of typify, the kind of compact, mundane thing that we think of as a finch. But concealed in that little mundane package, I think there's a lot of really cool biology. And I know many of you are familiar with it all already, but hopefully you all heard at least something new today and something to get you excited about finches. And I just want to put a shout out to Matt Young. I mentioned him before, who's the founder of the Finch Research Network, FERN. It is this thing that's dedicated to the study and conservation of finches and their habitats globally. And there's a lot that you can do if you become interested in this. You can share your expertise and sound recordings, your observations, banding data, pictures. There's places you can post these things. the recordings that are becoming available now through things like Xenocanto, eBird, and so forth are just they're a rich source of stuff that we can use for analyses, and it's really exciting. And there are a bunch of different projects Matt's got going. There's a thing he's calling the Crossbill project. There's an evening grosbeak project. We have automated recording units out at a bunch of different places in California and Oregon. This one, I'm afraid, may have burned up in the Dexter fire, but we'll see, to try to figure out where the boundary is between type 1 and type 2 evening grosbeaks. And I just want to finish by acknowledging my graduate students. This is one of our favorite capture locations, the sewage lagoons near Signal Mountain in Grand Teton, Mount Moran there in the background. This is Jamie Cornelius. There's Rod Kelsey, Kendra Sewell, who also I referred to during this talk. And I just want to encourage everybody to revel in doing nice, basic, descriptive, observational field biology. It's the foundation of everything that we could want to do, and it's extremely valuable. And I'll stop there. And if there are any questions, I will take them, and I'm going to end the show.
Yes, we do have a few questions.
Okay.
Two. If you have questions, please put them into the Q&A so that we can share them. And here we go. I've got the first question is from John Harris.
Okay.
He wants to know if the smaller finches also have large crops or gular pouches.
So pine siskins have a big, I don't know if it's physically as large as a crossbills, but they do have a big crop. They're lateral, and you can see it well on them. I haven't handled as many red poles. I don't know about red poles. So there's some of these I'm not sure. Maybe somebody else knows. But the gular pouches, like bullfinches, the gular sacs are in a few of the different birds, but not the sort of Carduelis finches and crossbills, and yeah, Cassin's finches have a crop. The Carpodacus, or Hemoris finches do as well.
All right, thank you. I have a question from Benjamin Sonnenberg. Does the mimicry imply that there is a very flexible crystallization period in finches, or possibly none at all? Do you know how much mimicry? Varies across geographic populations?
Yeah, this is a really good question. I'm going to comment first on your use of the term mimicry, which is what I've called it for years, but some evolutionary biologists get all hot and bothered about that because we don't know what the function is, and mimicry implies that it's like Batesian or Müllerian or whatever, some kind of formal mimicry. So I've shifted to calling it imitation, even though that has problems too, because we don't know exactly how it develops. Imitation implies a developmental, so does mimicry. But in any case, just think about that. I'm not criticizing you. I'm just, because it's heterospecific vocal mimicry is very prevalent as a term in the literature, but some people get irate about it if we call it that. I don't, but anyway, we don't have a lot of information yet about the geographic variation in how much of this is present. But what I can tell you is that it's very prevalent. All the places that I've found, for example, Cassin's finches. Cassin's finches' vocal note repertoire gets larger with age. Fred Sampson showed that, which is consistent with the idea that they keep learning new things that they could copy and add. But that doesn't rule out the possibility that they memorized it all when they were a baby and then just drew from memory when they added it in a subsequent year. So I think actually probably experiments need to be done on that. But those are challenging, obviously. So these are great questions. The answers are basically, I don't know, but it's getting recordings of these birds and looking at them and trying to figure out what all they are imitating, what stuff in there is copies of other species is really fun. So if any of you are into that, I encourage you to do it.
And we have a question from Lily. How did you find out that the crossbills were eating the insects?
Well, it was actually early. So it's been reported actually. There are old reports of it. If you look through old things like Gabrielson and Jewett's Birds of Oregon, they have all sorts of stomach contents, and they report significant amounts of insects in the stomachs of crossbills that they shot to put in museums. So it's not like it's brand new information, but what was really noticeable to me was when I was doing field work at the very beginning of my dissertation, and I was out looking for places where crossbills were going to settle and hopefully start breeding so that I could study them. In May and June, one of the first field seasons I did, they just weren't eating. I was finding crossbills, but they weren't eating any seeds from cones. In fact, a lot of them were in places where there were no cones. And I was watching what they were doing, and they were picking stuff off of the foliage tips. And a lot of that was these coolie gall aphids, which I didn't even know what they were. I collected some of it. I went back, and there was a graduate student at the University of Washington in the zoology department that I knew at the time who knew exactly what they were. And she told me all about these things. And then I started reading. I got a book about foliage insects. And so this is one of those things that I've been interested in for a long time. And I just think, and then one day, this was very opportunistic. I was walking into the library at the University of Washington, and I heard this just din of crossbill, sort of conversational song is what Matt Young calls it. It's just this cacophony of crossbill song coming down from an elm tree that was over the entrance to the library, and there were leaves raining down. And they were these elm leaves that had been curled up by these woolly aphids. And ran back and got my binoculars and was looking at these things and watching the crossbills, and they were picking the leaves off and opening these things up and licking them out and dropping them. And some of the ones they dropped still had aphids in them. And I had birds in captivity at the time. And I took some of those back and gave them to them. And they knew immediately what to do with these things. They just snarfed up these things. Oh, and they were good at scissoring the things open. So a lot of it was just sort of serendipity, accidental discovery. The whole thing about the petiole gall aphids was I was dropping my son off at a camp, and I camped overnight in a place up near Mount Shasta and found crossbills, and that's what they were eating. Jamie Cornelius has radio tracked a lot of crossbills in Wyoming, and there's sometimes when there are cones, they're focusing on insects. We spent like an hour one time trying to locate a male crossbill that had a radio on it up in this mixed spruce. It was blue spruce and cottonwood, and we're looking in the spruces because they're covered with cones. And finally, we found the thing in one of the cottonwoods eating aphids. That's what he was doing the whole time. So they eat a lot of insects, and I do think the insects are really important in helping them get to their new locations. That's conjecture on my part. I don't treat that as, gospel or anything, but I really think it's important.
Very good. Well, thank you so much for taking these questions. I am going to turn it over to John Harris now, and we're going to wrap things up for the evening.
Okay, thanks, Jen.
Thank you.
Well, thank you, Tom, and everyone, please join me in giving Tom a virtual ovation for an excellent talk. That wraps up our conference program for today. We'll reconvene tomorrow morning at 11 o'clock Pacific time. Our first talk tomorrow will be on California spotted owls and fire. So I hope you'll all be back for that talk, and we'll see you all tomorrow. And thanks again, Tom.
Thanks for inviting me, John, and thanks to everybody for coming in and listening.
Thank you and good night.
Bye-bye.
Good night.